All right. Okay, so yeah, I'm Marcia, and I'm going to talk about the reproducibility of in vivo electrophysiological measurements in mice. So this was one of the task forces at IBL. And the goal was really to address this major problem in the field, which is that understanding whole brain scale electrophysiological recordings requires the collective work of multiple labs. But the results in systems neuroscience are difficult to actually reproduce from different labs. So IBL was in a great position to address this problem and help because we're made up of many different labs, 10 different labs. And so these 10 different labs basically collected data, and we were able to use that to determine lab-to-lab reproducibility of electrophysiological results and standardize our protocols. So for this, the approach that we use is that we basically evaluated neural activities recorded from a repeated site. And that repeated site is shown here. These are the coordinates. And it basically passes through 5 different brain regions, the cortex, hippocampus, and thalamus, including VIS-A, AM, CA1, DG, LP, and PO. So yeah, so this work actually that I'm presenting here, it is online. It's published. So it's on bioRxiv, and it's been accepted for publication in eLife. We just got the reviews like a couple days ago. So you should be able to see that on eLife in a couple weeks as well. So you can look in there to get more information. But basically for this work, we had 121 sessions that were targeting the repeated site across the 10 labs. And we really standardized the experimental pipeline across the labs, everything from the head bar implant surgery all the way until the ex vivo imaging and the tracing and alignment. And we sent we perfused the brains, sent them for serial 2P microscopy to have a 3D reconstruction of the probe regions. And then we also used the probe track tracing lasagna to align these probes to different brain regions. And so on the right up here, you can see basically the 3D brain. And these lines here are where the probes were actually placed for this repeated site. So you can see there's actually quite a variability in where the probes were placed. And here you can see the coronal and the sagittal sections showing where those probes are. And then we wanted to really assess like this variability and quantify it between different labs because now that we have this histological reconstruction, we can know where the true probe trajectory is. And this is it's essential to know where these are located to interpret the recordings. So we compared a few different things. We had the planned recording, which is shown here in white. Then we had the micromanipulator location, which might be off from the planned one because of blood vessels in the brain or different skull features. And then we have the histology, which is like where the probe actually was. We also had to look at probe angle because this would determine where the probe was placed in the deeper regions of the brain. So I'm not going to go into details of this. This is just showing one of these analyses, an example. And just very briefly, we have the position of these probes compared to where we actually wanted the location to be. And so this is showing the difference, the distance between the histology and the planned coordinates. And we found that, there's a large insertion variance. And this, sorry, this limits our probe targeting specific brain regions. And we found that the resolution limit was about 360 micrometers, and this must be taken into account when planning the probe. Okay, so the major driver of this probe placement variance was really differences in skull landmarks used for establishing the coordinate system, and also differences in the underlying brain structures. Now, using our pipeline, we were able to, minimize this as much as possible, but there is still quite a variance that we have to keep in mind when doing recordings. Okay, so then we were interested, other than placement of the probes, we were interested to know if the electrophysiological features were reproducible between labs. So here, down here, you can see different probes from the different labs. Each color in these numbers indicates which lab it's from. And then the colors on the columns are showing which brain region the recording is from, the cortex, hippocampus, or thalamus. Each dot is a neuron, and the firing rate is indicated with the color of the dot. So, we were looking at these, but we wanted to know which of these recordings, which of these probes we actually want to include in our analyses. So which ones do pass our criteria. And so you can see on the flow chart here, we started out with about 121 insertions, but throughout, processing this, we decided to use some quality control metrics, and we ended up with 82 total probes to be analyzed. So, about 18 of these were just lost because we couldn't get histology from the brain, or there was some kind of hardware failure at the beginning. But the rest of them were mainly, we lost them because of the quality metrics, which I'm going to talk about in a little bit more detail. And then at the very end, we also lost 5 recordings because of the behavior. We didn't have enough behavior trials. Okay. So we wanted to, again, standardize some exclusion criteria for quality control, which is really this section on the top here. And we call this RIGOR, which stands for Recording Inclusion Guidelines for Optimizing Reproducibility. And I'm going to talk about that in a little bit more detail here, but generally it can be applied to a wide range of electrophysiological experiments. Some of the metrics might need to be adjusted for measurements in different animal types, different regions, or different behavioral contexts. Okay, so RIGOR standards consist of 10 different criteria. 7 of these criteria are related to the whole recording, and these are either computed automatically, which we have codes available for, or they have to be visually assessed. And I'm going to talk about that in a little bit more detail in a minute. But we have things, for example, the neuronal yield needs to be good enough, and then we have other things which I'll discuss in a moment. In addition to the whole recording criteria, we also have single unit recordings, or single unit criteria metrics, which include things like looking at the refractory period of each neuron. And again, there are codes available for all of this. Okay, so let's look at the visually assessments, the visual assessments of this criteria. So for example, here you can see on the top, we have the raster plot. And from the whole recording, the blue line indicates where the task ended, the decision-making task ended. And so, this recording up here passed, it looks beautiful. The recording on the bottom, though, did fail because, as you can see, there's a lot of epileptiform activity. So this is an example of where our visual assessment actually gets rid of some recordings. Another example here on the bottom is artifacts. So we have artifacts here, and so this recording also failed and we had to remove it. Another example here is drift. And finally, we can also look at the raw ePhys data. And so this is really zoomed in on a specific part of the task, and you can see it's kind of hard to actually see here, but in red and green, we're indicating the spikes. And on the top, these you can see that there's a lot of spikes visible, and so this passes our criteria. But the bottom one, there's not enough, and really these are noisy channels. So this is another example of a recording that needed to be removed. Okay, so again, we started out with 121 recordings. We ended up with 82 recordings that we were actually using for our analyses. And we had a total of 5,312 good single neurons that passed the RIGOR metrics, in the 5 repeated site brain regions that I discussed earlier. And so we wanted to see, now that we have this standardized method of really evaluating which recordings and which neurons to keep, do we have variability between labs? And again, I'm not going to go into all the details here. This heat map just shows kind of the summary of the results. So we're looking at the 5 different brain regions from the repeated site, and then we have 5 different electrophysiological features. And we found that for the most part, these features were reproducible across the laboratories. There's one exception here where there was a significant difference between the labs. Another thing that we did also do is we quantified, we did a quantitative assessment of the IBL data quality. So we compared our IBL data set with external electrophysiology data sets from both the Allen Institute and a paper from Nick Steinmetz. And we, made sure that we ran their data with IBL, pi kilosort version, as well as the parameters that we used. And when we did that, we found that in a lot of different aspects, which are kind of shown here, but especially as an example, looking at neuronal yield, the IBL data is comparable to the other data sets as well in all three brain regions. In addition to that, we also wanted to do a qualitative assessment of the data quality. And so I won't go into all the details, but basically we had three people who rated the different data sets without knowing where they're coming from by looking at the raw data, as well as, the raw data basically with the spikes overlaid. And then we found that it was basically the same. The quality was the same. Okay, so then we wanted to continue to understand more about how reproducible our data was between different labs in terms of functional neuronal activity. And we found that this was a lot more complicated than just the basic EFIS features. So it really depends on the brain region and the analysis method used. So here's an example. We have, so each color here is from one lab. It's the average, it's the recording average basically from all the cells recorded in that lab in all of the different sessions from LP. And these are all aligned to the movement onset. And so you can see, for example, here, the blue trace is from Berkeley, the lab at UC Berkeley. And we found that, there was a huge change in the firing rate of the neuron after moving onset compared to baseline. But in some recordings, that was not true. For example, here in green from UCLA, there was almost a 0 spike per second change in firing rate. So there's quite a lot of variability there. And when we quantified this, again, I'm not going to go into details, but when we quantified it in different ways and using different task modulation tests, and also we looked at, percent of modulated neurons and then the distribution of firing rates, we found that, there were quite a few areas where we did have significant difference between labs. And so this really tells us that single neuron analyses are very susceptible to a lack of reproducibility. And we looked at, sources for this, and it seems to be partly due to the high variability of single neuron responses. So we probably need very large sample sizes to mitigate this. We also looked at population activity using PCA, and we found the same results. This is just showing kind of the position of the good neurons that passed our rigor standards and where they are for the LP. And again, we found that the spatial position or spike waveforms, none of these really explain the variability we have between labs. So I'm kind of running out of time. So just very briefly, we set up a neural network model called the MTNN, and this to see if we can quantify any sources of variability. I won't go into the details, but overall, the big thing is that we found that behavioral, mainly really movement, was kind of the biggest source of variability. But overall, the lab and the sessions had close to 0 effect sizes. So overall, we were pretty reproducible in our results. So in conclusion, we observed notable variability in the position of the electrodes in the brain, despite our efforts to target the same exact location. After applying stringent quality control criteria, the rigor standards, we found that the EFIS features were reproducible across labs. The proportion of neurons that were modulated by behaviorally relevant task events and the ways in which they were modulated, that was more variable and more complicated. Our neural network model showed that lab ID accounted for little neural variance. Thus, the observed variability is mostly driven by outlier neurons, and having very large sample sizes is important. And finally, the standardization and quality control standards can enhance reproducibility. Still, caution should be taken with regard to electrode placement and their interpretation of some neural analyses. And a huge thanks to the entire IBL team, our task force, and all of the authors on the paper, and especially to our funders. All of the protocols, again, are available on our website. Thank you.